In this video, we're going to talk about how DNA gets copied. Remember that this happens in the S phase, the DNA synthesis phase of interphase. So in human cells, you have all of these long, stringy chromosomes. You've got 46 of them in each human cell, and they all need to be replicated. You need to have a copy of every single one before a cell can divide. So you have the chromosome is duplicated through the process of DNA replication. And after DNA replication happens, you have two sister chromatids joined together at a point called the centromere. But in this video, we're going to focus on what's happening here. So this is really the very basic idea behind DNA replication. So you have the DNA double helix with one strand and then the other strand joined together through base pairing and hydrogen bonding in the middle. What happens in DNA replication is that this double helix separates. It opens up, the two strands come apart, and then each parent strand serves as a template for the new daughter strand. Basically, each parent strand serves as a base, and then an enzyme comes along and builds a new strand complementary to the parent strand. The enzyme that does this, the major enzyme that does this, is called DNA polymerase, because it's an enzyme that polymerizes DNA nucleotides, puts them together. Now, DNA polymerase adds nucleotides to the new strand according to the rules of base pairing. So you have adenine pairing with thymine, and you have cytosine pairing with guanine. You can see that over here. You've got a guanine on the parent strand. DNA polymerase adds a cytosine on the new strand, on the daughter strand. Thymine on the parent strand, adenine on the daughter strand. By the time you're done, down here at the bottom, each new daughter DNA molecule is composed of one half a strand from the parent molecule, and the other strand is brand new. So DNA replication is often referred to as semi-conservative replication because it is half old molecule and half new molecule. This is actually important in terms of a cell being able to replicate its DNA without making errors. The fact that half of the original strand is still in there allows DNA polymerase to sort of tell which strand is the new strand and therefore which strand has the error if an error occurs. This is a bit more detailed a view of what's going on with DNA replication. You have the enzyme DNA polymerase here. You're also showing another important enzyme, helicase, over here. Helicase is the enzyme that actually unwinds the double helix. So helicase opens up the DNA double helix so that DNA polymerase can come in and build the new strands. There's a little bit of a twist to DNA replication that's illustrated by this diagram, and that's that DNA actually has a directionality to it. It has to do with how the nucleotides are joined together. So when you have the double helix, when you have two DNA strands joined together to make the double helix, they sit together in such a way that you have the 5' end of one strand facing the 3' end of the other strand, and vice versa. So it's like the two strands are lying head to tail. Now if I sketch that in on the diagram, You've got the 5' end of the parent strand there, and the 3' end of the parent strand up here. And then on this other parent strand, you've got the 3' end and then the 5' end at the end. Now, this is important in DNA replication because DNA polymerase can only add nucleotides at the 3' end of the growing new strand. So if we draw in the growing new strand, you have 5' on the new strand there, and then the 3' end is over there. But here you've got 5' there, and the 3' end on the new strand. So you have a DNA polymerase on each strand, building the new DNA molecule. But since DNA polymerase can only add at the 3' end of the molecule, DNA polymerase has to act a little bit differently on each strand. Helicase is moving in this direction, so that's all fine for this DNA polymerase. This DNA polymerase can move after helicase, just follow it down, adding nucleotides to the 3' end. This is called the leading strand. The strand can be built just continuously with DNA polymerase chasing helicase down. However, on this opposite strand, on the other side here, called the lagging strand, DNA polymerase can't chase helicase because it can only add nucleotides at the 3' end, so DNA polymerase has to move in a series of short little hops. It can add that segment and then wait for helicase to open up a new segment, hop backwards, add another segment. It has to move in a series of short hops. There are 3 major enzymes involved in DNA replication that I want you to be familiar with. I want you to understand what helicase does, of course understand what DNA polymerase does, and one more that we haven't talked about yet, primase. DNA polymerase needs a short RNA primer to basically tell it where to start making new DNA. And primase is the enzyme that lays down that short primer. It's about 10 nucleotides long. So on the leading strand, because it's synthesized continuously, the DNA polymerase just chasing down, you have a short RNA primer that primase lays down. DNA polymerase lands on it and starts building a new DNA strand. On the lagging strand, because it has to move in these short backwards hops, you have a series of RNA primers. So you've got one RNA primer for this segment, and when when helicase has moved on and DNA polymerase hops back a bit, you'll have another DNA primer laid down to start off, another RNA primer, laid down to start off the next segment. So think of that little short RNA primer as a docking point for DNA polymerase, as a signal for DNA polymerase to start building a new DNA molecule here. Alright, after studying this video, you should be able to answer these questions. Describe how DNA is replicated. Base pairing is very, very critical. Definitely explain the importance. You might want to explain why it's called semi-conservative replication as well. And you should also be able to explain the role of each of these enzymes.